So today we'll be discussing about another type of genetic exchange process which occurs in bacterial cells. So previously we had discussed about the conjugation process in bacteria. Now we'll be discussing the transformation method in bacteria. Okay, so basically transformation in bacteria is a method where genetic exchange happens in a way in which basically a naked DNA released by a bacteria who lyses or dies, it releases its naked DNA into the environment. And if there's another bacterial cell, so the other bacterial cell can take up this naked DNA and integrate this naked DNA into its own chromosome, into its own DNA, or yeah. So basically, what I'm saying is that transformation is a genetic exchange method in which and naked DNA. Now this is a thing to note, that this is the naked DNA, and it's very different. This process is very different from the conjugation process, in which the both bacterial cells were conjugated, and thus the plasmid was transferred from one cell to other. That was other story. But now this is a genetic exchange method in which the naked DNA. This is a real point to notice, that this is naked DNA and it's not within the cell. It's released by a cell which has died, in which naked DNA released by a dead or lysed cell is taken up by another bacteria. So basically, this naked DNA can either integrate to the new bacterial cell's DNA, or it can exist separately as a plasmid in that cell. Okay, so plasmid are basically extra chromosomal DNA which have their own machinery, and they divide by themselves, and they are basically an accessory uh genetic material in a cell. Sometimes they carry the resistance genes, which are important for bacterial resistance to certain antibiotics. Okay, so let me just explain with some examples and the origin of the transformation process, how it was discovered, and who did and what experiments he did. Okay, so in in 1928, there was a scientist with the name of Fredrick Griffith. Okay, and he was doing some experiments, and he was working basically with two strains of bacteria, two strains of Streptococcus pneumoniae. As you all have may have heard this name, this is the bacteria which causes pneumonia. So these are the Streptococcus pneumoniae, which cause pneumonia. So basically, Frederick Griffith was working with these two strains of Streptococcus pneumoniae. Okay, so one of the strain was the virulent one, meaning that it was pathogenic, and it had the virulence genes which are required to cause infection and virulence. Okay, so the one of the strain was the virulent one, and the second strain was the avirulent one, meaning not virulent one, not virulent or pathogenic. like they don't carry the pathogenic genes. Okay, so virulent strain of Streptococcus pneumonia basically had a structure called capsule. Capsule is a structure found in many bacteria which help them to prevent phagocytosis by the host cells or eukaryotic cells. Okay, so these are the cause of these virulent strain of Streptococcus pneumoniae. And so the A virulent strain of Streptococcus pneumoniae lacked capsule. And that's the reason that they are not virulent and they can be readily phagocytized by the host cell. Okay, so Griffith was basically what was the aim of Griffith? Why was he working with these two strains? So basically, Griffith's aim was to determine whether uh, injections of heat-killed bacteria of the encapsulated strains would be used. Can they be used to vaccinate mice against pneumonia? So it was basically his aim, and as he expected. So what will happen? Like let me just rub this. Okay, so basically, so Griffith's aim was to develop vaccine against pneumonia. Okay, so basically, there was a living encapsulated bacteria. He conducted an experiment in which they were there were living encapsulated bacteria used strains of S. pneumoniae, and second was the dead encapsulated bacteria, and third were live, living, unencapsulated bacteria, basically non-encapsulated. Sorry. And yeah, that's it. So basically, what did he do in this experiment? What Griffith did is that, um, first of all, he gave injections. Mouse were used in this experiment. So first, what did is that he gave injections of live encapsulated bacteria. And just imagine what will happen if living encapsulated bacteria are incorporated into the mice. This was the first step I'm discussing. Let me just show it and erase these one. Okay. So what will happen when live encapsulated bacteria are incorporated into the mice? What will happen is that into the mouse. What will happen? Mouse will Mouse died. Okay, so first he did this. He incorporated living encapsulated bacteria into the mouse, and the mouse died. Okay, and then second, what he did is that when he gave injection of live non-encapsulated bacteria or dead encapsulated bacteria, they both didn't kill the mice. So what happened? Live, but they are not capsulated, non-encapsulated, and dead. They are dead but encapsulated, meaning that they have the capsule. When these were integrated, when their injection was given to the mouse, what happened? So basically, they stayed alive. They stayed alive and healthy. And the reason for this is that, so basically, if the bacteria are, so just note that in this step, the live non-encapsulated bacteria are separately given to the mice, and the dead encapsulated bacteria are given separately in another step to the mice. So what happens is that when a separate injection of live encapsulated bacteria is given to the mouse, so basically the bacterial cells are basically live, but they don't have the capsules, meaning that they are not virulent, so they don't cause the infection, and the mouse remained healthy. And the same for dead encapsulated is that the cells are these bacteria is dead, but it has a capsule. So basically, the capsule is of no use if the bacteria is dead. So the mouse will not die; it will stay healthy and alive. Okay, so now the what he did next is something very, it basically the basic, you can say the principle of the transformation process. This step is where the transformation was first discovered. So what happened is that in the third step, basically, the live but unencapsulated bacteria glass. So this is a thing to note that the injection has both the live unencapsulated bacteria, sorry, and it also has dead but encapsulated bacteria. Sorry, encapsulated bacteria. Okay, so what will happen when they are introduced into the mouse? What will happen is that you must guess that what will happen. So basically, what happened was very surprising that the mouse died. Okay, so this is very surprising because the injection had the live unencapsulated bacteria, which was meant to be avirulent, and the dead encapsulated bacteria, which was also meant to be avirulent. So what happened was surprising for Griffith, and this was the basic introduction to the topic of transformation, which was discovered then. So basically, many discoveries were made, and hence the conclusion was that. So basically, what happened is that the capsule of dead bacteria had made the live bacteria encapsulated. So basically, in simple words, this is what is the basic principle of transformation. So what happened was that the capsules of dead bacteria had uh, entered basically you can say that the DNA of the dead bacteria, the naked DNA. Remember what I said in the beginning that the naked DNA had been taken up by another bacterial cell, and it had become integrated into the cell's DNA. Okay, so this is what happened. That is why the live bacteria became, which were initially not containing capsules, they were later containing the capsules. That is why the mice died. Okay, so let me explain with the diagram so it will be easy. Okay, so just suppose that this is a bacterial cell Okay, and this is the DNA of it. And just suppose that it has the capsule genes, you can say capsule genes. It has the genes which code for the capsule. So it had the this cell has the genes for the capsule, which is why this bacteria is capsulated. Okay, so just suppose that this is a cell, and just suppose that this is another cell with it present, like it's in close proximity. Okay, and just suppose that this cell has its own DNA, but this DNA does not have capsule genes. So if this bacteria dies, if the cell dies or lyses, what will happen next? What will happen is that this cell will release its DNA into the environment. It will release its naked DNA into the environment, which will then be taken up by this cell. This is the competent cell, which competent basically means that the cell is the most in a state, it is in a physiological state in which it can take up the DNA very easily, and basically its cell membrane is very permeable. So it has permeable membrane at this point, at this state. So this is why it takes up this DNA. And basically, what will this DNA do? Now, let me just draw this cell again. Okay, so let me just first erase. Okay, so what will happen is that, just suppose this is the competent cell I was talking about. Okay, and now what has happened? Just suppose that this is its own DNA, and just suppose that the red one is the dead cell's naked DNA, which has been integrated with this DNA. Okay, okay, so this is the and as I have mentioned that this part of the DNA had the capsule genes, so which is why this cell, and just suppose that this was initially the live unencapsulated bacteria, but now it has transformed to live encapsulated bacteria with the capsule genes, which was initially of the dead bacteria's DNA, but now the dead bacterial cell has released its DNA, and that DNA has been integrated with this DNA. Okay, so this is the transformation process. Just note that either the naked DNA can become integrated into the uh, this bacterial cell's DNA, but it can also exist as a separate plasmid isolated from this DNA. Okay, so that's another case. Okay, so this is the transformation process, and so basically, this transformation process gave rise to the inference that DNA are genetic material. It basically says that the DNA is the main genetic material which was transferred from one cell to another. DNA is the genetic material and the basis for this genetic exchange mechanism. Okay. Okay. So basically, what I had discussed um about the all these steps of transformation. Let me just mention them here so it's easy for you to remember them. Sorry, transformation, not transportation. Okay, so let me just mention them here quickly. So, first step is the release of DNA by the dead cell. Second step is the uptake of DNA by another cell. Third step is the integration of DNA with new cells' DNA. And the last step of transformation is the expression of new traits, in this case, the encapsulation of the live bacteria. I can say that expression of encapsulation by living bacteria. Okay, so this was about the transformation method in bacteria, and just to be clear, that transformation is a type of horizontal gene transfer, and this is a common genetic exchange mechanism which happens between two bacterial cells. And so, if you like know about the other genetic exchange mechanisms, it will be easy for you to remember about this one as well. And that's it.